so now we are going to talk about the DNA, what the DNA is made of. We'll be also talking about DNA and RNA. Basically, we will be seeing what this DNA is made of. So DNA is basically a polymer. So polymers are long chains which are made of monomers. So monomer is a subunit of a polymer. So DNA is like a polymer, it's a long chain, and it is called a polynucleotide chain. So poly means multiple, nucleotide is the monomer. So the DNA is made of nucleotides. So we'll see the structure of the nucleotide, what nucleotide is, and we will then see the differences, what are the differences between DNA and RNA. So each nucleotide has three components, and what are those three components? It is made of a pentose sugar. Pentose means 5. It's a sugar which is made of 5 carbons, and it has a nitrogenous base with it, and it also has phosphate group. So nucleotide is made of three things: a pentose sugar, nitrogenous base, and phosphate group. I'm going to talk to you about the details of this nitrogenous base and also the phosphate group. But if there is no phosphate group, then it is not called a nucleotide, then it is called a nucleoside. So if pentose sugar and with the pentose sugar nitrogenous base is attached, then it's called a nucleoside. So you can see here, this is the structure of a nucleotide. This is a nucleotide. So you can see here, this is a pentose sugar, a sugar with 5 carbons. So it's a kind of a ring structure. It has 5 carbons. Carbon number 1 here, 2 here, 3 here, 4 here, and on 4 is attached the number 5. On carbon number 1 is attached the nitrogenous base. And what is the nitrogenous base? These are the nitrogenous bases. So nitrogenous bases are ring structures. They are made of carbons and nitrogens in them. And they are of two kinds, purines and pyrimidines. Purines are adenine and guanine. So these are the two kinds of purines, adenine and guanine. And pyrimidines are cytosine and thymine. And in DNA, the nitrogenous bases which are present are adenine and guanine, cytosine and thymine. And this is the first difference. In RNA, instead of thymine, there is another nitrogenous base called uracil. So first of all, let's see why they're called nitrogenous bases. They're called nitrogenous bases because they are ring structures. You can see that these are all ring structures, and they have nitrogens in them. You can see these are ring structures made of carbon and nitrogen. So you can see carbon and nitrogen, carbon and nitrogen. So they are ring structures which are made of carbon and nitrogen. And you can see, okay, another question is why they're called bases, why they're called nitrogenous bases. They're called nitrogenous bases because when you add them into water, the pH becomes alkaline. So that's why they're called the nitrogenous bases. And these nitrogenous bases, they are attached here on the carbon number 1 on this ring structure. So this is the carbon number 1. This is called 1 prime, this is called 2 prime, this is called 3 prime. Then here is a 4 prime, and on 4 prime is attached the 5 prime. Now why these carbons are labeled as 1 prime, 2 prime, 3 prime, 4 prime and 5 prime? They are labeled as prime because you can see these are the ring structures. These are the nitrogenous bases. They also have, you can see they also have the carbons in them. Because the carbons in these nitrogenous bases, they are labeled as 1, 2, 3, 4. Just to distinguish the carbons which are present in the nitrogenous bases and the carbons which are present in this sugar, just to distinguish them, they are labeled as 1 prime, 2 prime, because the carbons in these rings, they are labeled as 1, 2, 3, 4. Just to make, just to differentiate, these carbons are labeled as 1 prime, 2 prime, 3 prime. Okay, so the nitrogenous bases are attached here. In DNA, the nitrogenous bases are adenine, guanine, cytosine and thymine. In RNA, there is no thymine, but instead there is uracil. And this is carbon number 2 here. This is carbon number 3, 3 prime, 2 prime, 3 prime, 4 prime, and on 4 prime is attached the 5 prime. On 5 prime carbon is attached the phosphate group in a nucleotide. So phosphate group is here, and this is negatively charged. Now both DNA and RNA, they have pentose sugar, which means the carbon with 5 the sugar with 5 carbons, But here lies the main difference, another difference between DNA and RNA. The sugar which is present in DNA has H here, and the sugar which is present in RNA has a OH here. So the sugar which is present in DNA is called deoxyribose sugar, and the sugar which is present in RNA, it is called the ribose sugar. It's called deoxy. It has one oxygen less, and you can see here on carbon number 2 prime, there is H, but on RNA it is OH. So in DNA the sugar has one less oxygen. So this is this sugar, and on 3 prime there is always OH group. It is same in both DNA and RNA. On 3 prime there is a OH group. So these are the nitrogenous bases, these are the sugars, and this is the nucleotide. Nucleotide has a pentose sugar, nitrogenous base, and a phosphate group. So this is the basic structure of the monomer of DNA or RNA. I explained you both. In RNA there will be uracil here instead of thymine, and also the sugar will be having OH, and in DNA it will be H. Third difference between DNA and RNA is DNA is double stranded and single stranded. So DNA is two strands which are wound around each other, but RNA is a single strand. So you can see here, and now this is a nucleotide of DNA. Okay, now how these nucleotides form a long chain. Okay, so that's what I'm going to tell you. Now this is your first nucleotide, this is your second nucleotide, this is third, this is fourth. Now how this nucleotide is going to be linked with this, how this is going to be linked with this, only then it is going to form a polymer, which is in DNA it is called a polynucleotide chain, and polynucleotide chain is made of a monomer called nucleotide. This is this nucleotide. Now this nucleotide and this nucleotide, at the 3 prime end here there is a OH group, and 5 prime end here is a phosphate group. So OH from this nucleotide and phosphate from this nucleotide, they together lose one molecule of water, and when they lose one molecule of water, that results in the formation of a covalent bond between them, and that bond in DNA is called a phosphodiester bond. So this is the way these monomers, they link with each other to form a polymer. So in DNA the monomers are called nucleotides, and they link with each other by a covalent bond here, and it is called a phosphodiester bond, and which links these monomers, which link these nucleotides, and it links these nucleotides and it forms a polymer, and the polymer is the polynucleotide chain, which is the DNA. Now you can see here, if the nucleotides are oriented like this, so this way on this side is 5 prime. So this is the nucleotide, this is the 5 prime, this is the 3 prime, this is a 5 prime, this is a 3 prime, 5 prime and 3 prime. So this chain forms one strand of DNA, and remember that DNA is a double stranded, there are two strands. Now in this strand of DNA, in one chain of DNA, you can see all the phosphates, the phosphates are on 5 prime and they are on this side. So 5 prime of this nucleotide is here, it is here, it is here, it is here, and OH is on this side. So you can see in every nucleotide 5 prime is on the upper side and 3 prime is the lower side. So for this chain which forms the one strand of DNA, this end is called 5 prime end and this end is called 3 prime end. So this is the way, this is the one chain of DNA, and DNA is double stranded. Okay, DNA is double stranded. There are two chains, two strands of DNA which are wound around each other. For these two strands of DNA, these two chains of DNA, they are anti-parallel in nature, which means that if this is one strand, and in this one strand 5 prime is on this side and 3 prime is on this side, for the opposite strand 3 prime will be on this side and 5 prime will be on this side. So the two strands or two chains of DNA, they are anti-parallel in nature. If the 5 prime is on this side for this chain, 3 prime will be on this side. So for the opposite chain, if this is the 5 prime, it's going to be 3 prime, and if this is the 3 prime, it's going to be 5 prime. So the two chains of DNA, they are anti-parallel in nature, which means that if this is one strand of DNA, in the opposite strand this nucleotide will be flipped. will be flipped so that the 5 primes of the second chain, they will be on this side, and all the 3 prime hydroxides will be on this side. So the two chains are, they are antiparallel in nature. So we have seen what DNA is made of. It is made of a nucleotide, and we have seen what are the 3 components of the nucleotide, and we have seen what a nucleoside is, and we have seen the structure of a nucleotide. We have studied all these nitrogenous bases. They are ring. Purines are adenine and guanine, and these are double rings, the single rings. These are sugars in DNA and RNA, and we have seen how these nucleotides, they are linked with each other, and the two chains are antiparallel in nature. Okay, we have talked about all these things. So we have talked about differences in DNA and RNA also. So another important thing to remember is, now these are the 2 strands of DNA. Now the 2 strands of DNA, they are wound around each other, and these 2 strands, they are linked with each other. The 2 strands of DNA, they are linked with each other. Now how they are linked with each other? They are linked with each other by complementary base pairing rules. And what are those complementary base pairing rules? According to A always links with T, C always links with G. And how they are linked with each other? They are linked with each other through hydrogen bonds. A always links with T, C always links with G. So this is the way 2 strands of DNA, they are linked with each other. The 2 strands of DNA, they are linked with each other through the nitrogenous bases. And there are rules. They are called complementary base pairing rules, according to which A always links with T, C always links with G. This is the basic structure of DNA. The backbone of DNA, which is when I talked about, when we were talking about how one nucleotide is attached to the other nucleotide, is attached by covalent bonding, and covalent bonds are very, very strong bonds. And the name of covalent bonds in DNA is phosphodiester bonds. They are very strong bonds, and they form the backbone of DNA. And the true strands of DNA, they are linked to each other by hydrogen bonds, and they are the weakest bonds. And this is the RNA. RNA is single stranded, but at some points there can be some sort of looping in the RNA, and in that looping there can be 2 nitrogen bases can link with each other, but RNA is always single stranded. And this DNA is basically a chemical which makes our genes, and our genes are present on our chromosomes. So our chromosomes, they have a lot of genes, and these genes are made of DNA basically. And DNA is a very, very long molecule. And how does it gets accommodated inside the nucleus? Because nucleus is very small, it is wrapped around the proteins called histone proteins. And if the cell is not dividing, then this DNA will be wrapped very, very tightly around these proteins, and it will look like a very, very dense mass, and it is called chromatin. And when the cell is actually dividing, this chromatin gets condensed and it takes the shape of the chromosomes. So in the nucleus, DNA is organized into discrete units called chromosomes, but these chromosomes will be visible only when the cell is dividing. Each chromosome is composed of single DNA molecule associated with proteins, and basically DNA wrapped on proteins in a non-dividing cell is like a dense mass, and it is chromosome, and this chromatin condenses to take the shape of chromosomes. And nucleolus is a region inside the nucleus where rRNA is made, and there are basically 3 kinds of RNAs. They are mRNA, messenger RNA, rRNA, and tRNA. We will talk about all these things. rRNA is basically a component of a ribosome, and ribosome is a protein factory where the proteins are made, and this ribosome itself is made of ribosomal RNA and proteins. And basically there are 3 kinds of RNAs. There's a mRNA which carries the message of the DNA, which is the which is the basically a king. mRNA is basically carrying the message of DNA, and RNA is a component of RNA is a component of ribosomes.